In the old days when I was doing this, I just looked at the audience and say, raise your hands if you can't hear me, but it doesn't work with Zoom. So anyway, to start over again, the first thing to do is to thank Aga and Travis both for their administrative and their technical skills in keeping these presentations going. It's absolutely spectacular to be able to continue sound learnings in the face of this pandemic. I also like to thank the audience for coming to these presentations. We've had an excellent turnout, and I hope you enjoy today's presentation. We don't need coordinators with Zoom, so the dangerous part of this is that the speakers get to introduce themselves. I'm a retired professor. I spent 39 years on the faculty at the University of Michigan. I taught undergraduates and I taught graduate students. I mentored a number, a number of graduate students in their PhD thesis research, and I also trained some postdoctoral fellows. Our field of research in the molecular biosciences basically was concerned with using the techniques of chemistry, biochemistry, and molecular biology to understand certain aspects of the reactions in photosynthesis that allow plants and algae to convert water into oxygen. So without further ado, let's talk about genetic engineering, genetically modified organisms, CRISPRs, and the future of human health. So the first thing is to have some definitions, and I'll get to those in a minute. Then we'll talk about GMOs briefly, the economics, the regulation of GMO formation, the benefits, health and nutrition, and there's still objections, and we'll talk about those. CRISPRs, what the heck are CRISPRs? CRISPRs are, you'll see this a couple of times, clustered, regularly interspaced palindromic repeats. You don't have to memorize that, but you do have to know that this is really made for a revolution in the life sciences. We'll talk about the discovery. We'll talk about CRISPRs and how the gene editors work. We'll talk about new molecular medicine using CRISPR gene therapies, and I'll give you some examples there, a couple of examples. And there'll be some conclusions, and there'll be time, as Aga said, for questions at the end. So keep in mind, if you have questions, think what they might be, and we can bring them up and talk about them at the end. So the definition of a GMO, first of all, any GMO is an organism that's got new genetic material that's been added to it or has been modified in it using genetic engineering techniques. So what's genetic engineering? Genetic engineering is a toolbox. It's analogous to screwdrivers and saws and hammers and all that sort of stuff, but it's a series of molecular techniques that scientists can use to move a gene, that is DNA, between organisms. You can move DNA between plants and bacteria, between bacteria and plants. You can move genes in just about any way that you want to now, in such a way as to bring DNA that wasn't there before, into an organism for experimental purposes or for basically purposes of medicine and human health. The new CRISPR technology, which we'll talk about, adds a new toolbox, and this technology is a technology that allows people to modify an existing gene in an organism without moving any DNA anywhere. You can go into an organism and modify that organism's DNA. Or you can add a new gene. You can make a GMO using CRISPR, and it turns out that almost all GMOs now are being made using the CRISPR technology. Okay, so some other definitions. These are things I think most of you know a lot of this already. Genes are DNA. Remember, DNA is a hereditary molecule. That's the fancy word. It's also the ancestry molecule. Many of you probably had your DNA done by Ancestry.com or 23andMe. Humans have about 20,000 genes. Bacteria have 1,000 genes. So you can go to bed tonight feeling really good that there are 19,000 genes between you and an E. coli bacterium. Genes are made up of the genetic code. We won't go into this in any depth, but there are these three-letter words in DNA. You see them sometimes on television screens as somebody's trying to tell you something about this gene or that gene or DNA. And these three-letter code words are A, T, C, Gs, and each one is unique for an amino acid and a protein, except for 4 words. And those 4 words say, 1, start. It tells everybody in the whole world at the molecular level, here is where a gene starts. And there are 3 that say stop. They say, this is where the gene ends. So every protein in the whole world, starting with you and everybody else down to bacteria, the proteins start life with a gene, a letter that says ATG, and that's for the amino acid methionine, or in bacteria, a chemically modified form of methionine. So that's something you know now that you can take to cocktail hour. Proteins, as you know, are the molecules of life. They're structural, they muscle, skin, hair. They're enzymes that accelerate biological reactions. They carry oxygen like hemoglobin. And importantly, nowadays, they protect us through antibodies that are proteins that are actually able to attack foreign proteins and prevent them from hurting us. So the last thing, and this is, what biologists like to be very important and very ponderous, is that there's the central dogma, but it's useful to illustrate things. The central dogma was set down years ago, and the central dogma basically says that the DNA code is copied into an RNA code, and an RNA code is then translated into amino acid sequences that makes the proteins that make our lives go round. And in a picture, this is the central dogma. DNA is double-stranded. It's replicated every time a cell divides. So your DNA goes with every cell division to the next cell, and that's why you accumulated 20,000 genes over a huge number of ancestors. And that code and the DNA stays basically not immovable, but sort of immutable in the cell and stands as sort of the source of information. But day to day in the cell, you have to make proteins, you have to get things moving, and that's where this process called transcription takes place, and you make RNA. And RNA is a slightly modified form of DNA, and it can be used in the cell and be read so that a little machine called a ribosome is able to make protein out of the RNA code that came from your DNA code. Now, the neat thing about RNA is that RNA doesn't stick around for a long time. RNAs are very sensitive, and so they get broken down. And this is one of the important things in biology, is that there's control, that you can switch a signal on by making the RNA, but it won't persist in the cell. It'll be broken down, it'll be stopped, it'll have to be made again, and this is the way cells control what's going on. But in the end, you wind up with proteins. So when we're talking about making genetically modified organisms, we're really talking about taking a piece of DNA, moving it into another organism. What we're really doing is we're moving in a new protein. Okay, so this kind of research really got going in the mid-1970s, and it's exploded. It's taken over almost all of the life sciences, and it all resides in the ability to manipulate DNA, to do bioengineering, and to change genes and to swap genes and to go on and on and on like that. I tried to find numbers just so you'd have a feel about the economy and what's going on here. On the commercial side, this would be big pharma, companies like Merck, Johnson & Johnson, and so on, Pfizer. In terms of human resources, it's estimated there must be at least about 450,000 jobs in that sector of the economy, with an annual income which is now greater. I put the greater in because of the pandemic, greater than $112 billion. And the tax revenues are about 15 billion or so. If you added to that all of the jobs in molecular biology that have to do with DNA engineering that are taking place in hospitals and universities and so on, I think you could probably add another 75,000 jobs or so. The annual income would go up dramatically. About tax revenues, I'm not so sure, because universities like the one I work for found pretty good ways of dodging the tax collector when it came time to pay up in that respect. Okay, so a lot of genetic engineering now goes on. It's widespread, but it doesn't go on unregulated. And I want you to know a little bit about that. And the watchdogs in this case in the government are the FDA, the EPA, and the USDA. They all maintain review boards in Washington or Bethesda, Maryland. And universities themselves have to maintain review boards so that anybody that wants to do any manufacturing of GMOs, wants to do any engineering of DNA, has to provide information about the experiments they're going to do, what they're going to do, and it has to be approved, sometimes all the way up to the federal level in Washington. And some of the questions that get asked are questions like this. If you're going to transfer a gene from one organism to another, is the gene itself safe? I mean, genes are sneaky things. Sometimes they can get away from you. There are things called transposons that jump around, for example. Is the gene going to behave itself through your experiments, and is it going to be restricted in its where it's being put and how it's going to behave? And the second question that's always asked, is there going to be any threat to health or food security? You have to assure people that that's something that's not going to happen. There are going to be no threats. And third, you have to be able to assure the people in the review boards that there are methods in place to control the spread of the GMO. Let's say it's a bacteria. You have to be able to assure people that that bacterium is not going to get loose in the lab. For example, when we were doing these kinds of experiments, we were transferring a photosynthetic protein from spinach into a bacterium. There was no problem with the health or food security because people eat, several tons of spinach every day in their salads and so forth and so on. But we had to be absolutely certain that we could present a convincing argument that we control the spread of the GMO if it got loose. And the way you do that is that you pick a way in which to grow your recipient of the gene, like in this case, E. coli, under conditions where it can't possibly live anywhere else. And that just meant using a growth medium that had some exotic nutrients in it that weren't available anywhere else in the environment. And with that, we got okayed, and for about 10 or 12 years, we were able to do these experiments. But almost annually, we had to report on what we were doing, and we had to assure people that we hadn't changed any of the conditions that we'd set forth. So this is regulated, and there's, there are always, these sort of science fiction terror things about wild organisms getting loose and everything. That's not going to happen in the case of generation of GMOs by bioengineering. So having done all this, what's the outcome? So let's look first of all at some GMOs that are now pretty much old hat, but weren't at the time they were first made, and their impacts on human health. So the oldest that I'm aware of, GMO, in the human health field is the production of insulin in bacteria for the treatment of diabetes. This was Eli Lilly Laboratories in Indianapolis, and it's in the 1980s, the early 1980s actually, that they managed to get the genes for insulin into bacteria and to produce the hormone in that way. And what that did in effect is made insulin more widely available and relieved Eli Lilly and other manufacturers from having to go to slaughterhouses and get pancreatic tissue and mash it all up and isolate insulin from that tissue. And in those cases, there are actually some problems occasionally with immune responses to the isolated insulin. So this kind of set the stage. It said, we can do this. What else can we do? And I've selected a few. One is clotting factors to cure hemophilia, or not cure it, but alleviate hemophilia. These clotting factors used to come from isolated blood banks where you had to take serum and actually isolate the clotting factors there. Now it's done by having hamster cells express the gene for the particular clotting factor. And I want to assure you that there are no large numbers of hamsters being slaughtered to do this. The hamster cells were probably harvested from a couple of hamsters way back when, and these have been kept in liquid culture, and they have been genetically modified with appropriate genes to produce the clotting factors. So you can use bacteria, you can use mammalian cells. One of my favorites is a drug called atrin, which comes from goat's milk. This is an anticoagulant. People have an overactive clotting system. The anticoagulant helps to get around that. And in this case, the drug company that developed this actually made genetically modified goats who have the gene for the protein, the atrin protein, and that is directed into the milk. So they milk the goats and then they isolate the drug from the milk of the goats. Humira, which is advertised widely on television. Almost every night I see a Humira commercial. somewhere, and this comes from mouse cells, and this is used, it's an antibody, I think, that's used in combating various inflammatory diseases. And back to bacteria, Neulasta from bacteria. This is a protein that's used to positively affect white blood cell production. In particular, I think it's applied or given to people that are undergoing certain types of chemotherapy. Now there's one more, and I want to deviate a little bit from the path I'm on to talk about this, because it's not exactly a GMO, and it's not exactly DNA, but it's something that you all know about, and I just want to make sure that you're all up to date on what's going on there. And that's the production of human antibodies by injecting RNA into human beings, and that's used for COVID prevention. So you know about the drugs, the vaccines from both Pfizer and from Moderna, and you've probably heard that you're injecting RNA. So let's look at what's going on here to kind of finish off this aspect of GMOs in human health. So you've all seen this, I'm sure. This is the COVID virus, and it's the target of the RNA vaccine. It is not red and gray. If it had a color, it would probably be like tan. The brown, or the gray part in there is a membrane, and inside that membrane is the virus's genetic material, which happens to be, in this case, RNA. And on the outside, these red things are the so-called spike protein. So this comes from pictures that can be gotten using electron microscopy. You have to have very high resolution, and the resolution is so good that actually you can get the structure of the spike protein itself. So let's mark the spike protein. Now, the nasty thing about COVID-19 is that if you start counting, and I've tried, I come up with about 50, at least 50 different spike proteins on this particular virus. And these spike proteins have a very high affinity for binding to certain types of human cells. In fact, they like to bind to angiotensin converting enzyme proteins. That's ACE proteins. Some of you may be taking a drug to help control blood pressure because these are called ACE proteins. And we have a lot of them. And the virus is loaded for bear because it has at least 50 different chances to bind to a human cell. And when I say binding, I'm thinking of something pretty much like an interaction, a Velcro interaction. It can be reversed, you can pull it off, but it goes on and it really sticks tightly. And so you have a target here for an RNA vaccine, and it's this spike protein. And foreign proteins in our body are called antigens, and our immune systems make antibodies against these proteins. So the spike protein is a foreign protein. It would be an antigen. And if you could get an antibody to react with these spikes, you would effectively prevent the spikes from binding to human cells, and you would be able to prevent infection by the COVID virus. So let's talk just a little bit about this. This is kind of a side issue, but it's something that I think is interesting and has some relevance today. So RNA is the code in COVID-19. You've got an RNA virus, and the spike protein is what attaches the virus to our cells. And when the virus does that, it enables the membrane of the virus to fuse with our cell membranes, and the virus gets into the cells. And once that RNA is in the cell, then the RNA code gets copied. It takes control of protein synthesis, and this is the way viruses operate. They're parasites. It's the only way they can operate. COVID-19 then makes new viruses. This isn't good for the cells or anything else. Our immune system will kick in and begin to attack it, but sometimes even our immune system overreacts, and we have these problems that come up with the infection. So Pfizer and Moderna both prepared vaccines which are now in active application that are based on the RNA code for the spike protein. And the way this works is that the RNA is produced in the lab, in a factory setting actually. It has the complete code for the spike protein, and it's prepared in an injectable form that's called, euphemistically, a delivery system. And the delivery system happens to be a collection of some lipids, a few sugars, and a little bit of a derivative is actually antifreeze. And when this is all shaken up in the right way, what you wind up with are these globes of lipids with the RNA trapped inside, and the presence of various protectants are there, so that in the case of Pfizer, you have to freeze at minus 80 degrees centigrade to keep things happy. It's not quite that cold for the Moderna vaccine. And this delivery system can be injected into you. Many of you have probably had the injection already. And those little globules of lipid carrying the RNA will find human cells. The lipid will stick to the lipids of the membrane. And once it's stuck there, the RNA of the spike protein code is delivered into the cell. And the cell says, oh, RNA, well, let's make some protein. And the protein it makes is the spike protein. And the spike protein is an antigen. And that antigen then turns on our immune system, and we make antibodies against the spike protein, which effectively blocks the RNA virus from being able to attack our cells. And that's the basis of the whole thing. There are several, our cells produce antibodies against the spike protein, provide the immunity, and so on. I've heard several things going around that I'd like to talk about because there's some mistrust of this particular antibody system, and I'd like to just address them head on. The things that I've heard, the first is that the vaccine causes COVID infection. It can't. The vaccine is an RNA coding for the spike protein and only the spike protein. There's nothing else there. So you don't have any of the genetic information that's required to make a new COVID-19 virus. You just have the information to make the antigen that's going to make the antibodies that's going to protect you. Secondly, the way these RNAs, and in the case of the J&J and the AstraZeneca, they use DNA, but in all cases, these RNAs or DNAs are constructed in such a way that they cannot get into our genetic system and interfere with that at all. So there is no alteration of DNA. And in fact, the RNA in these RNA viruses, I told you that RNA is sensitive. It's kind of unstable. Once you're inoculated and you get the RNA in your cells, it will last probably a week, maybe up to 10 days. And in that time, it will serve as a message to make the antigen that allows you to get the antibodies. But the RNA is going to be destroyed. So it's not going to hang around for a very long time, and you're not going to be a wandering RNA factory for the spike protein. And the last thing, which really is an item of concern even among some healthcare workers, is that the perception is that these vaccines were prepared too fast. And that because they were prepared too fast, there's something wrong with them. Well, let's think about this for a minute. With most of the vaccines we're familiar with, and let's take polio and measles as cases, Jonas Salk needed 3 to 4 years from the start to develop a vaccine against polio, and John Enders at Letter Labs needed up to 9 years, I think, to prepare the vaccine against measles. In both cases, they had to do this because they did not have the molecular biology toolbox. The only thing they had reference to was a way of using the virus itself as the antigen, which means that you have to go through a whole series of very important steps to make sure that the vaccine itself is inactivated in such a way that it can be injected and serve as an antigen, but not as an infective agent, okay? And there were cases in the development of the polio vaccine, we know with the Cutter laboratory incident where, in fact, somebody slipped up and there were infections from some lots of the early polio vaccines. This is why it took so long to make most of the antibodies we're familiar with. Using the molecular methods that I've just told you about, you skip everything having to do with having to treat the virus this way, that way, and so forth and so on, and testing with monkeys and testing with volunteers and what have you. And you go straight to the antigen, you get the code for the antigen is expressed in test volunteers, 40 or 50,000 people, no ill effects, and that's basically the way that we're now getting we're getting our vaccines. And I think this is a good thing because basically we'll probably be looking at other pandemics down the road, and we now have a very rigorous system for dealing with this. So those are kind of the myths about these RNA vaccines, and I wanted to just talk about that for a minute. And if you have questions, there'll be questions at the end of the talk. You can ask for more if I know the answers. So this is sort of the overall picture of what's going on with human health and GMOs and the use of molecular techniques. Let's talk about human nutrition for just a second, because in case of human nutrition, there's a huge impact, as a lot of it is directed at strategies that will improve crop yields. And I'll come back to this in a second again. There are a lot of herbicide-resistant crops out there. A lot of them resist Roundup, and this is for weed control, and that gets up crop yields. Insect-resistant crops, they use the Bacillus thuringiensis toxin, and that's for insect control. They are now rushing as fast as they can to develop drought-resistant plants, and they want to, in this case, offset the effects of global warming, reduced rain, and they're already succeeding with certain types of plants. And there's a fourth one that's interesting, and that is the vitamin A enriched or golden rice. This rice, rich in vitamin A, prevents childhood blindness in developing countries where vitamin A in the diet is just not sufficient to help with the development of the visual system. And so plants, GMO plants really have a role, not only, I guess you'd say it's human health, but it's human nutrition. And this is very important. So let's put a summary of these things that we've gone over so far. In the case of human health, we have increased drug supplies now. In many cases, we actually have drugs you couldn't get before because they were so rare. They were so low in concentration, it was hard to get them from any biological source. And in fact, this gives us access to unique drugs that are now available that we couldn't get before, including some antibodies that just were hard to develop, that can now be done all the way by using genetically modified organisms. And this leads to new therapeutic strategies with regard to, for example, clotting diseases, erythrocyte production, and so on. In the case of human nutrition, the important thing here is that the race is on to increase crop yields to beat what's called the 2050 problem. The 2050 problem is that it's been calculated that around about 2050, the growth in the human population will exceed the growth in crop production, and this will mean mass starvation. And so there are projects, a number of places. The one that is probably the best in the United States is at the University of Illinois, where they have had success in turning the photosynthetic process around and increasing its efficiency so that they can get more protein, more crop per acre, using molecular techniques instead of the standard hybridization techniques. But this is a real worry and something that USDA here and various funding agencies in other countries are now working on. Improved nutrition, protein content, in particular, healthier plants because of warding off disease, and the use of lower toxicity herbicides. Now that's the pros. There are the cons as well. In the case of human health, there's a science denial streak out there in the population that just says that foreign DNA is bad. You can't do it. You shouldn't do anything with it. You shouldn't monkey with the natural law and so forth and so on. And I don't know of any argument you can use to set aside that argument other than to point out that there's nothing bad about DNA and about what's being done with it. Another argument is that DNA transfer is harmful. This is the idea: you get DNA in the wrong organism, you're going to get some sort of a Frankenstein monster. And we just went through all of the hoops that researchers have to jump through in order to be able to do these experiments, and it's very obvious that that's not going to be something that's going to happen. And there is a concern in some cases that there could be allergic responses to GMO proteins. As far as I know, this has not happened yet, but it's something that has to be tested and has to be looked at in certain cases, and that's in this whole regulatory scheme. Human nutrition, you can throw in all the points above, and you go to human nutrition, and herbicide-resistant weeds have come up. They can be controlled, but that's a problem. Anti-GMO food labeling is now very popular. You can go over to Publix if you've got half an hour and wander up and down the shelves, and you will find out that, much to your amazement, there are such things as genetically modified oranges and genetically modified tomatoes and so on, where in fact there are no such organisms at all. There have been tomatoes, but that flopped a long time ago. It is basically sort of a scare technique, and I think that the people behind that plot are the people that eat organic, but I can't prove it. The last thing is Roundup causes cancer. This is an issue right now. The science says it doesn't on the basis of National Cancer Institute study of 55,000 agricultural workers. The 3 juries in San Francisco say that Roundup does cause cancer, and without much scientific input, the company Bayer Monsanto is now going to settle up, and they will pass the cost of that settlement on to the farmers, who will pass it on to the grocery stores, and so we'll be paying a few cents more for food in the future. Okay, so let's take the situation then, and I picked 2016 for a reason, that in general in the United States and more globally in Europe even, gene transfer regulations have been firmly established, and everybody seems to play by the rules. The techniques, the toolboxes, and the organisms are available, and they're used for all sorts of gene transfers. Yeah, drug production is one that I've emphasized here because I think it's one that has the greatest good in terms of human health. And the same thing in agriculture and nutrition, with increasing the nutritional value of crops, trying to keep the prices down, trying to use the least toxic herbicides. And the United States does really lead the world in biotechnology research and development. There has been, I think, an enlightened view of this kind of research here that says with proper monitoring, no harm will come from it, and it should be given its lead. There are countries, France and England, for example, which are very, very strict, and where there are real problems in trying to deal or grow any kind of a genetically modified organism. So the one thing that was missing, and here we go, the missing technique was that nobody had a toolbox that allowed them to go straight into an organism, find a gene in that organism, and modify it. And this is where the CRISPRs come in, clustered regularly interspaced short palindromic repeats. That's the last time I'll use that. It was the 2015 Scientific Discovery of the Year. It was on the cover of Time magazine. CNN was bubbling about it, just on and on and on and on and on. Now, this just didn't pop out of nowhere because somebody, some man or woman in a white lab coat with horn-rimmed glasses had a eureka moment. This was a slug it out in the trenches sort of thing, and I want to tell you a little bit about the history of CRISPRs when we talk about them. So this goes back probably 10 to 15 years at least, and it started with people that just study those words, those 3-letter words, the codes in DNA. And you do that because out of that, you begin to learn how DNA gets turned on and turned off, and you get to find places where maybe there's damage or something's happened to the DNA, and that might be affecting the organism. And so it's a very serious kind of thing, and it's big science. You use computers to do this because there are literally, millions and millions and millions of these 3-letter words in the genetic codes of the various organisms being studied. In this case, it was bacterial DNA. And what was odd about it was that there were these palindromic repeats, and I'll show you one in just a second, whose sequences are identical to the DNA that's in viruses that attack the bacteria, not in the DNA sequences of the bacteria itself. So the question is, what the heck is a virus sequence doing in the DNA of a bacteria? Palindromic sequences, this is one. DNA runs in two directions from its 5' end to its 3' end, and that just means that there's a phosphate on the 5' end and a phosphate on the 5' end. And if you look at this, if the red is red, if you read the red forward and you read the green backward, they both read the same thing. That's a palindromic sequence. And it turned out that when they hit upon these regions in the bacterial DNA, where there were these palindromic DNA sequences, that's where these viral virus sequences were also clustered and hiding. Now, when people saw this, they began to scratch their heads and trying to think, what could this possibly be? And one idea that went around was the notion that maybe this somehow reflected the history of the bacterial DNA, that this bacterium had been insulted or attacked by a virus, and that in having this DNA from the virus there, it actually gave it some kind of immunity against viral attack. Well, the other thing they found was that near all these CRISPR genes in the bacterial DNA, there was always a gene for a protein called Cas, and that means CRISPR associated. And the most popular one of these was called Cas9. There are a lot of Cas proteins, 3, 7, whatever. But Cas9 kept popping up and nobody knew what it did. And this is pretty unusual in an era where if you have a protein and you have a DNA sequence, you can usually look at it and begin to think, well, that looks like some kind of this enzyme or that enzyme. It just didn't fit. So again, a lot of conjecture and a lot of guessing and a lot of speculation. And the answer came, I think, probably from several labs, but my favorite is the following. Scientists at the Dannon Yogurt Research Laboratories in Copenhagen were having a serious problem with some of their prize bacterial colonies. They use bacteria to make yogurt, and the yogurt, the bread and butter for Dannon. And what they found was that some bacteria were really sensitive to the viruses, and others weren't bothered at all. And so, chance favors the prepared mind. There were scientists at Dannon that were also thinking about the problems with these palindromic sequences and with CRISPRs and everything else. So what you do, because it's relatively fast, is you just do a complete DNA sequence of the particular organism. And lo and behold, what they found was that the bacteria that were protected against virus attack all had these palindromic CRISPR sequences, and the bacteria that were being wiped out by the virus didn't have the sequences. Bingo, this is the aha moment. Then there was a scramble back to the lab because now you got to figure out what is going on here. And I can tell you, and then I'll show you a cartoon. So it turns out that protection against the virus occurs in a bacteria when a small piece of RNA gets copied from CRISPR DNA sequences, and the CRISPR DNA sequence, lo and behold, interacts with the Cas9 protein and steers the Cas9 protein to the matching DNA sequence that's released from an invading virus. And Cas9 is what's called the nuclease, lo and behold. And Cas9 cuts the viral DNA and inactivates it and protects the bacteria. So following the discovery of all this, there were all kinds of cartoons and pictures and everything else about how CRISPRs work. My favorite is this one. Okay, so those two blue strands are a piece of DNA from the virus, and it's being unwound by an interaction with that yellow stuff, which is a piece of RNA from a CRISPR sequence in the bacterium. And that sort of chartreuse creature from the Black Lagoon that's lurking behind is Cas9. And Cas9 is up there leaning over, and it's going to whack that viral DNA, and it's going to make a cut in it. and that cut is essentially an inactivating event. So this is an artist rendition, but it's probably pretty close to what it actually looks like in bacterial cells and in any cells where you have CRISPR activity. That the Cas9 can be directed specifically by the RNA sequence to a particular place in the DNA where it cuts. There's a line drawing here, and you don't have to look at anything except where it says cleavage on that gray kind of glob there, because what that illustrates is that this guide RNA actually brings the Cas9 enzyme right in to a very specific spot in the DNA where it will hit right at one particular letter in a genetic code. And the cleavage, it will break it right there. Highly specific. Excuse me, you can make the break, and then there are arrows below. You can make that break, and then you can do other things, which would be, you can repair the break. So let's say it was a mutant of some kind, or you can insert new DNA and make a GMO. And so bingo, this solves a major mystery, and this opens the door to a whole new generation of molecular biology experiments. So applications. There are two people who really got things rolling. One is Jennifer Doudna, and the other is Emmanuel Charpentier. They're both at Berkeley at the time, working together, and they developed a CRISPR-Cas9 system, basically a toolbox, had the right enzymes, had the right RNA and DNA and everything, that allowed people to go in and edit DNA in bacteria. And in fact, this got to the point where you could actually, high school kids in biology classes were doing CRISPR experiments on yeast, and this was like 2 years ago. So it's a really simple thing, but it was very clever discoveries. A great deal of intellectual effort went into this. So right on the tail of this came a person named Feng Zhang. He was a professor at MIT, and he developed the same type of system for animal cells. Now, you got bacteria on the one hand, you got animal systems on the other hand, and you got Berkeley on one coast and MIT on the other coast, and so that means you're going to have a fight over patents. That happened over a period of a couple of years. Berkeley got the bacterial patents, and MIT/Harvard, the Broad Institute, got the animal patents. There really weren't any hard feelings because since that time, Doudna, Charpentier, and Feng have cooperated together in developing various CRISPR systems for DNA editing. Jennifer Doudna has written a book. She's given TED Talks, and she basically has now started her own biotech company, aside from her work as a faculty member at Berkeley. Emmanuel Charpentier is now the director of one of these very important scientific research centers in Germany. So everything came out nicely in the end. There's been a lot of progress. So the question is, what's so great about CRISPRs? Let's sort of summarize what this does. First of all, it's a much more efficient way to create GMOs by DNA insertion. I can testify to that myself. In the old days, using the old methods, it took us 6 months to get a protein out of spinach, a photosynthetic protein out of spinach, and get it into a bacteria where we could get lots of the protein to study. 6 months. It would have taken us about 10 days using the CRISPR technology. You can specifically target a site of DNA insertion when you're making a GMO. You can use it to block a gene you don't want. You can cut right in the middle of that gene and put something else in and know it goes there. You don't have to fiddle around with all kinds of assays and testing. Afterwards, one quick sequencing of the DNA and you're off and running. And like I say, it's extremely efficient. It requires much less time to create a GMO. You can also edit an organism's DNA. It is possible to go into an organism with the CRISPR system and to look for, example, damaged genes. That would be mutations. As I showed you in that line drawing, you could make a cut right in the middle of a mutation and you could do a repair job by insertion of the correct sequence. Another thing, and I'll give you an example of this in just a little bit, is to do what's called gene silencing. That is, there are genes that get turned on and they shouldn't be turned on. They should be off, and you can silence genes just by doing it, just by cutting them someplace, or by fiddling with their sequence a little bit and shutting them down. Okay, there are ethical issues, and I think many of you are probably going to be familiar with some of this. It is appropriate only to use CRISPR technology if it is not carried out on eggs, sperm, or embryos. That's totally prohibited. And I think many of you will remember that a couple of years ago, there were these stories about the twin babies in China that had been created using the CRISPR technology. The doctor who said he did it is now in jail, and there's been nothing further about these children. And I've seen the papers that supposedly describe what was done to do the treatments and everything, and it looks like a bunch of mumbo jumbo to me. So I think this may have been a case of fake science. I'm not sure that this was actually done. You can use CRISPR technology to correct genetic issues, but it's permitted only if the experimental material is made up of mature cells, somatic cells. So no sperm, no eggs. Okay, so there's a huge literature out there speculating about the use of CRISPR to target various genetically linked diseases. This would include Huntington's chorea, Lou Gehrig's disease. There are a number of diseases where proteins are known to be involved in the causative phase of these diseases. And if they're proteins, that means there's a gene somewhere. That means that if you can find the gene, you could do treatments. None of those, as far as I know, have reached the experimental stage yet. But I want to talk about ones that have. There's an inherited blood disease. It's called beta thalassemia. The patients that have this can obtain inefficient oxygen if it's by their hemoglobin, because there is a mutation in the hemoglobin itself that interferes with oxygen binding. You have to have blood transfusions to get normal hemoglobin so that you can get the appropriate blood levels in your bloodstream. Okay, so the people that have that particular problem with the mutation essentially have to have regular blood transfusions. If they don't, oxygen transport is critically important, and you can't have that with beta thalassemia. The mutations are in the hemoglobin, and a lot of people have looked at that thinking that there might be a way of correcting the problem in the proteins that make up hemoglobin. It turns out it's just too complex. The other problem is in sickle cell anemia. This is a blood disease again. In this case, it's well known what it is. It's been studied in a number of populations around the equator, around the whole world. In each case, it turns out that a big amino acid called glutamate with a negative charge has been changed by mutation into a valine, which is a tiny amino acid which has no charge on it. And this mutation causes big structural changes in the hemoglobin protein. So what happens in these patients is that they suffer intense pain when their blood cells adopt a shape that's like a disc. And that's because of the structural change in the hemoglobin. And for these patients, just to basically exist and to survive, again, they need blood transfusions. And it's a fairly terrible thing. So this is a case where the scientists that are working on, the medical scientists, the molecular biologists, are thinking outside the box. Here's what's been done, and there are trials, and I can talk about them just a little bit. Right. Here we go. Okay, so this is the potential therapy. Cells from these patients are edited using the CRISPR technology, and it's used for gene silencing. What it does is it inactivates a gene that blocks the formation of a protein that normally inhibits the synthesis of something that we all have, and it's called fetal hemoglobin. Talk about that in just a little bit. So we have fetal hemoglobin when we're developing before birth because we're competing with our mothers for oxygen, and fetal hemoglobin basically is a standard hemoglobin, but it is able to bind oxygen more efficiently than adult hemoglobin. And it can replace the function of adult hemoglobin. In fact, when we are born, we still have fetal hemoglobin, but it's basically shut down progressively by this protein from this gene, this protein that inhibits the synthesis of fetal hemoglobin because our adult hemoglobin is kicking in. So as of late 2020, and I just looked this up in late December when I was putting this together, both sickle cell anemia and beta thalassemia patients in small trials, and I think this is up to maybe 20, 25 people, were all doing very well after receiving CRISPR-treated cells that synthesize fetal hemoglobin. So what's being done in this case for these patients, they take some bone marrow cells, they CRISPRize them, if you will, to shut down this protein. They put the cells back into the patient, and lo and behold, fetal hemoglobin appears, and oxygen transport, normal oxygen transport is restored after this particular treatment. So these are the two studies I know of that have actually gone for some time now. They've been approved, and the studies now, not large numbers like testing vaccines and things, but a fair number of people, and they all seem to be doing well after being treated with these particular cells. COVID-19 diagnosis, I had to throw this in at the end. A biotech firm that belongs to Jennifer Doudna just reported that they have a new technique for detecting the COVID-19 virus using CRISPR. And in this particular case, the new technique uses CRISPR methodology. It detects the virus in infected people. They say it takes about 5 minutes total from beginning to end. And so if this hits the market, it's a new way of tracing COVID, and may help us along the way to stamping it out. Okay, so in conclusion then, what's going to happen because of all this is that GMOs in the future will all probably be produced using the CRISPR technique. It's too specific, it's really fast, it's ideal for doing this kind of work. But more importantly, there are potential therapies that are very promising. Excuse me, and there will be more trials that are needed. And one of the things that people will have to look at carefully is the cost. I have heard that for some of the work that's proposed to be done, if it were carried out, the cost per patient would be over $1 million. And that's not something that can be easily sustained. And again, this is a case where it's a one case only, and there's no mass production or anything else. The cost could come down there, but that's still very expensive. The no eggs, no sperm, no embryos rule is being enforced. As far as I know, that is worldwide. And so there's no tinkering with heritability at this point, although it's something that's going to be debated, and it may eventually happen that it will be the target of CRISPR therapy. And the CRISPR technique for gene editing is the most significant recent advance in the life sciences. There is one more thing that you ought to know. There's the 2020 Nobel Prize in Chemistry, and it went for CRISPRs, and that's Jennifer Doudna, and that's Emmanuelle Charpentier. It's the first time 2 women have gotten a Nobel Prize, let alone a Nobel Prize in chemistry, and it's a richly deserved honor. So that's what I have. I'd be happy to answer any questions.
All right, ladies and gentlemen, if you do have a question, Mrs. Carol has a question. Go ahead and unmute yourself.
This is Bob Rutan with my wife Carol. One thing I've heard about the potential COVID vaccine, what happens in, say, 2026 when the coronavirus of that year comes along? Will there be a interaction or some potential problem? Thank you.
That's a good question, Bob. What I suspect, because it's already going on, we're talking essentially about the appearance of these variants, which are changes in the sequence of the spike protein. I think that the vaccine manufacturers are going to be able to stay on top of that all the way along, so that either by way of a booster shot or if it's somebody's first shot, the vaccine that will be given will be one that will account for any changes in the spike protein amino acid sequence that could have occurred by mutation. I'm thinking right now that basically both Moderna and Pfizer are actually probably working on some sort of a mixed RNA vaccine that contains RNAs that have the sequences of the one we're familiar with now and the sequences of the variants as well as something that can be given in the future. That's a guess, but I'm thinking that, given the way these people think and the way they work, that's something that's probably happening.
Ladies and gentlemen, I just launched our poll. I promised I was going to do at the end of our session. Please take your time before we finish this seminar to give us your opinion about the subject discussed. And we're still open to questions. Go ahead and ask them. This is the time to do that. In the meantime, Mr. Jochen, do you think that the vaccine is going to be approved to use in children at some point?
Oh yeah, for sure. They just haven't been able to carry out the tests on children to the extent that they want to before they okay it for use. If anything, it would be adjusting the size of the dose based on the age of the child. Young children, smaller children might not need as much of the vaccine as, say, an 18 or a 19-year-old. Maybe those kinds of things.